Hey everyone, welcome back to Windows Heat Studies. I hope you all are doing well. So we started discussing about immunology, where we first learned about what is immunity and what are different types of immune system that our body possess. Then we started discussing about the cells of immune system, the different types of cells that are found in the body, in the immune system, that are part of innate immunity as well as the adaptive immunity. Right. So we discussed about the process of hematopoiesis, means the process of blood formation of mature blood cells from the hematopoietic stem cells. So we discussed that in detail, and there we learned about different types of the cells that are part of our immune system and how they are formed, they are differentiated from the hematopoietic stem cells. So today we are going to learn about natural killer cells, which are a part of lymphoid lineage, okay, and they are different from B lymphocytes and T lymphocytes because they are a part of innate immune system, unlike those. So hematopoietic stem cells, just to have a recap, hematopoietic stem cells, they get differentiated into myeloid lineage as well as lymphoid lineage, right? The lymphoid lineage has the lymphocytes, the cells that belong to lymphoid lineage, they are referred as lymphocytes, okay? And they are B cells, the B lymphocytes, T cells, T lymphocytes, and there's one more category known as natural killer cells, which will I'll be writing as NK cells. Okay, so those natural killer cells, they belong to lymphoid lineage, but unlike B cells and T cells, they are a part of innate immune system. B cells and T cells, they express the antigen specific receptors over their surfaces, and that is why they are specific. And being specific makes them a part of adaptive immunity because they have the capability of adapting themselves according to the experiences that the individual is being exposed to over its lifespan. But unlike them, natural killer cells show no expression of such antigen specific receptors, and that is why their way of functioning is very non-specific, and that is why they are a part of innate immune system, just like the cells that belong to the myeloid lineage, right? So we have been discussing about neutrophils, eosinophils, basophils. We have already discussed about the other cells, dendritic cells, and now as well as macrophages. So today we are going to discuss about natural killer cells in detail, how they get activated, what are the different mechanisms of killing by the natural killer cells, so how they are going to clear the pathogen from the body of the individual, as well as we'll also learn about some assays to measure the activity of natural killer cells. So this is the topic of focus today. So let's get started with the lecture. So natural killer cells, as I told you, they are lymphocytes, and previously in the introductory lecture, we discussed that lymphocytes, they are agranular. They do not have granules. But the natural killer cells, on the other hand, they have large granules, and that is why they are also referred as granular lymphocytes. Okay, so the granules of natural killer cells have different enzymes as well as different proteins that are going to help them in the killing the target cell which is being infected, and that is why the granule composition is again important in the case of natural killer cells. So the granules, they have the proteins such as perforins and enzymes such as granzymes. Okay, so these are the components of granules, and I'll be discussing them in a while when we will discuss about the mechanism of killing the target cells by the natural killer cells. Okay, so for now, just remember that these are the enzymes and the proteins that are going to help in killing the target cell. So they are going to help natural killer cells to kill the target cells, the cells which have been infected due to some virus. Okay, whenever a virus infects any cell, the last resort that our body has to do, and the last job that it has, is to kill that cell, because for limiting the virus, we cannot directly kill the virus, right? So for limiting the viral replication becomes important. So whenever the virus infects the cell, killing that cell is the best way to get rid of that infection. So killing those target cell which has been infected with those pathogens, that is done by the natural killer cells. And for that, they doesn't really need activation, means they don't need any, the antigen specificity, and that is why they are part of the innate immunity, right? So these are these specialized cells which will destroy the virally infected cells. So they are specialized, okay, natural killer cells. Let me write it here. Natural killer cells, they are specialized in killing the virally infected cells. They can also kill the tumor cells because, well, they are abnormal and they are going to cause the damage to the individual, right? So every day, almost every day in your cells, they are undergoing some replication, right? So they are replicating, they are dividing. So there are chances of them mutating and turning into some abnormal cells, some tumor cells. And in your body, your cells, they are undergoing such abnormal mutations. So such cells will be killed even before the tumor forms in your body. So the cancerous cells or the potentially dangerous cells, they will be killed by the natural killer cells, okay? And when the that is why the evasion of immune system by the tumor cells plays a very important role in the tumor, in the formation of malignant tumor, because this is how tumor cells are going to trick your immune system into believing that they are harmless, when in reality they are very harmful. So this is how the immune cell does not only take has the job to kill the pathogen, to kill the whatever foreign particle that is going to cause the damage, but it also kills the cells of your own body that will be problematic in future, right? So, and natural killer cells, they also kill tumor cells, and they are also helpful in killing the stressed cells, okay, the cells which have been exposed to some stress and they are turned into abnormal. So natural killer cells, they are, they have those specialized jobs. So they are part of innate immunity because they do not need any prior sensitization like the B cells or T cells, right? So they do not have this antigen specific activation need for them to work, because, well, they have granules, and upon the degranulation they can simply kill the target cell. So there are different types of the receptors that are present over the natural killer cells, which helps us in the recognition of the natural killer cells. So those are known as surface markers. So those surface receptors which are present over the natural killer cells, they are very specific, and I'm going to list them down here for you. So let's discuss about surface markers of natural killer cells. Okay, so there are different surface markers such as CD56. Okay, so this is also referred as NCAM because it is neural cell adhesion molecule. And this particular receptor is very important because it is going to define the subset of natural killer cells. So there exist some regulatory natural killer cells as well as some cells which are going to be highly cytotoxic. So there are different types of the natural killer cells depending upon what job they do. So for recognition of those different types, this surface marker helps a lot. So this surface marker kind of defines the subset, okay, subset of natural killer cells. So this helps in defining that subset. Then there is other surface markers such as CD16, and it is actually a receptor for the antibody IgG. Okay, so this is actually FC gamma receptor 3. Okay, it is also referred as FC gamma receptor 3, and it binds to the FC region of antibody IgG. Okay, so this particular receptor is going to help into antibody dependent cellular cytotoxicity. I'll come to this particular topic in a while. So for now, just it is important to know for antibody mediated killing of target cell. For that, that is also a job of natural killer cells, and that will be mediated by this particular receptor. Okay, and besides those receptors, there are some other receptors present on the natural killer cells, such as NKG2D, which are helpful in activating receptors for stress induced signals, right? Because the stress cells needs to be killed, and that will be helpful when the stress ligands are released. And for receiving those stress ligands, there are some receptor, and for activation of such receptors, the NKG2D receptors are going to help. Okay, so these are the few surface markers that are present over the natural killer cells. So let's now discuss about the different types of natural killer cells. There are two main types of natural killer cells. The first type is very, it is highly cytotoxic, but it has, the cytokine production by this subtype is low. Whereas other subtype, the other natural killer cells, they are, they have high cytokine production, but cytotoxicity is low. Okay, so for identification of those subtypes, we need the help of surface markers, the CD56 and CD16. So the cell subtype which has high cytotoxicity and low cytokine production, they have low amount of CD56 over them. And that is why it is referred as CD56 dim, and abundance of CD16 marker. Okay, so both of those marker are present, but CD56 marker is present in low abundance. The expression of this marker is less, whereas the CD16 marker is quite abundant. So such cell types are they are highly cytotoxic, means they are going to kill the infected cells as well as the tumor cells. So they take participation in killing, but they produce low cytokines. So they are not really good regulators. They are not really good at activating other immune cells. Okay, so this is the result of the cell flow cytometry, and that is why we read those cells such as CD56 dim, because during the flow cytometric analysis, the intensity of color is very less, and that is why we refer them as dim, because the CD56 expression is less by those cells, whereas they do express CD16. On the contrary, the other type, they has higher expression of CD56, and that is why we refer it as CD56 bright. And the CD16, it could be present there, but it is very down regulated, so it is in very low amounts. So we refer it as the CD16 minus, because there could be no expression of CD16, or it is down regulated, means it is so low that it is unrecognizable, it is negligible. So this has higher expression of CD56 and lower expression of CD16. So the first type, this first particular type, which has low expression of CD56, it is going to act as the commanders, because it is regulating other natural killer cells with the oh sorry the first type, which is highly cytotoxic, it is going to act as the frontline commanders, frontline fighters, means they are going to actually they are going to actually participate in the killing. Whereas the second type, it is working like a commander, Means it is going to regulate the killing. It is going to it is going to mediate the cell signaling, and it is going to result into the giving out or handing out the roles to the particular cells that they have to kill the target cell now. So they are more regulatory natural killer cells. So they are also referred as regulatory natural killer cells because of the high cytokine production, whereas these cells, they are referred as the cytotoxic natural killer cells because they are actually going to do the job of killing. Okay, whereas these are only handing out commands, they are only giving, so they are only assigning the roles to the other natural killer cells as well as other immune cells besides the natural killer cell, such as macrophages or neutrophils. Okay, so those are the two subtypes of natural killer cells, this is how, using surface marker, we can define both of them. So for besides, so here I discussed a few surface markers, okay, CD56 and CD16. So these are for recognition. But besides those receptors, there are other receptors which are going to result into either activation or inhibition of natural killer cells. For example, one of them I discussed here, the NKG2D. So this is actually the activatory receptor, means it is going to result into activation of natural killer cells. But besides this activating receptors, there are other inhibitory receptors which are going to result into inhibition of natural killer cells present over the surface of those cells. Why? Because as I told you previously, for you to reach to certain location, your vehicle needs to have an accelerator as well as brakes, because you need to stop at certain point of time, right? So you need to increase your speed to reach to that location, but you also need to apply brakes so that you can stop there and you can actually say that, okay, I have reached to my goal, I have reached to my destination. So in the same way, our immune system needs to have accelerators, means it needs some activatory proteins, activatory receptors over it, as well as it needs some inhibitory receptors over it. So this balance between activatory receptor and inhibitory receptor, whenever it is disturbed, at that point of time, the natural killer cells will get activated. Means let's say that the expression of or the activation of activated receptor is high. So this is going to result into activation of natural killer cells, mediating the killing job. And if the inhibitory receptors, they are activated, they are high, means the brakes are applied. So now the natural killer cells, they won't be able to they won't be able to work. So the different types of receptors that are found there, the activatory receptors, I have already discussed one of them, the NK2GD receptor, and as well as the CD16 receptor, because it is also going to mediate the antibody dependent cellular cytotoxicity, right? So both of them, they are the activatory receptor. On the contrary, there exist some inhibitory receptors such as KIR, which is killer cell immunoglobulin like receptors. So these are killer cell immunoglobulin like receptors. Okay, so this receptors, they are inhibitory, means the activation of this receptor is going to inhibit the natural killer cells. So what happens is that the natural killer cells, so as I discussed previously, there exist two types of MHC molecule, MHC1 and MHC2. So MHC molecule, they are present in the cells of an individual, all of the cells which have nucleus. So only the RBCs which do not have nucleus are the cells which are not going to express this MHC1 molecules. So MHC1 molecules, they kind of act as the flag to, or a label which is labeling those particular cells that this belong to that particular individual. So let's say that there are two individuals, the person A and person B. Okay, both of them, all of the cells belonging to person A and person B, all of them, they are going to have MHC molecules, MHC type 1 molecules. Okay, but the MHC type 1 molecule belonging to person A, those will be different from the MHC type 1 molecule which belongs to person B. And that kind of serves as an identity for those cells. So they serve as the flag which is giving them the identity to the cell, that this cell belongs to this particular person, right? So that is why the natural killer cells, they are going to only, they need to kill the target cells which are non-self, means which does not belong to this particular individual, and that is why they need to sense MHC1 receptors, right? So the signal that is read by natural killer cells, that is referred as the missing self cell signal. Okay, it is referred as missing self signal or missing self recognition. Okay, I'll just explain it why it is referred as missing self signal. So what happens is that this receptors, this killer cell immunoglobulin like receptors which are present over the natural killer cells, they recognize the MHC class 1 molecules. Okay, so whenever those gets activated due to the recognition, due to the binding to MHC1 class molecules, they are going to inhibit the natural killer cells, because they are like, wait, stop, this is our own cell, so we don't need to kill that, right? So whenever this cell does not bind to this receptor, does not bind to MHC class 1 molecule, means a cell, the target cell does not have this MHC class 1 molecule, so this the particular receptor is not binding to that MHC class 1 molecule, means it is missing MHC class 1. So this signal, this particular signal is known as missing self signal, means the absence of MHC1 molecule is going to give the missing self signal to the natural killer cell, which is also referred as missing self recognition. Because, okay, so there is, non-self recognition and there is missing self recognition. Let's learn about the difference first. Okay, so there is two, non-self recognition and missing self recognition. So missing self recognition will be done by the natural killer cells, whereas non-self recognition simply means that uh, you know recognizing the antigenic particles, means the particles which are foreign to the body, whereas missing self recognition means recognizing that I don't know whether to whose like to which type of class of molecules this particular molecule belongs, but I certainly know that this is not a self molecule. Okay, I hope you're getting the difference between non-self and missing self. Non-self simply means that this particular cell type or this particular antigen is foreign. Okay, so this particular antigen is foreign antigen. That is known as non-self recognition. Whereas missing self recognition means the antigen is non-self, means it does not belong to, or no, not antigen is non-self. Missing self recognition, sorry, missing self recognition means that the self antigen is missing, right? If the self antigen is missing, means that particular cell is either non-self cell or it is a self cell but a dangerous cell or a damaged cell. Okay, so that is why missing cell recognition is very important by the natural killer cells, because and this they both of them, non self recognition and missing self recognition, is very different, because in the non self recognition you are literally, this is only going to recognize the cells which are being affected by virus, the cells which are being affected by the bacteria, but this won't be able to recognize the tumor cells, because tumor cells basically they are your own cells. They do have MHC1 molecule expression, but that expression profile is unusual, and that is why only non-self cells will be recognized by the non-self recognition. But the missing self recognition by natural killer cell is very very important, because it is actually sensing the self antigen missing, means it is sensing the absence of self antigen. So absence of self antigen could mean that the cell is non-self, means it does not belong to that individual's body, or the cell does belong to that individual's body, but it has unusual profile of the MHC1 molecules. And whenever a cell has an unusual profile of the MHC molecule expression, MHC molecule 1 expression, this simply means that even if that cell belongs to me, but that cell is damaged and it needs to be killed because it is going to cause damage, further damage in future. And that is why the missing self recognition, this property by the natural killer cells, this is used in the immunotherapies, in the cancer treatments for training the natural killer cells against the tumor cells. Okay, so this is very important. So missing the natural killer cells, they are going to do the missing self recognition with the help of certain receptors such as those, and that is going to result into the natural killer cell activation. Okay, so whenever natural killer cells gets the missing self signal, it is going to get activated. But if it is binding this particular receptors, those are binding to MHC class 1 molecules, this means that particular cell is our own cell, so that is going to result into formation of the inhibitory signals, and the natural killer cell won't get activated. Okay, so this is how the balance between the activatory receptor and the inhibitory receptor is very important for activation of the natural killer cell. So how does natural killer cell gets activated? So it gets activated due to different chemical mediators such as interferon gamma, IL-2, interleukin 12, IL-18. So these are the few chemical mediators that can activate the natural killer cells. So the mediators that can activate natural killer cells are interferon gamma, IL-2, IL-12, and IL-18. So these are the ones which are going to activate the natural killer cell. So once the natural killer cell is activated, it has different mechanisms for killing. It can opt any of the mechanisms for killing the target cell. So I'm going to discuss about those mechanisms that natural killer cell follows for killing those target cell. So either it could directly do the cytotoxicity using those releasing those perforin or granzymes. So it could be direct cytotoxicity, which is mediated by the degranulation, and the degranulation will release perforin. Okay, perforin, which is a pore forming protein, which is going to form pores, and it will eventually kill the it will eventually kill the pathogen. And it will also release granzymes, the enzymes which are these are actually proteases. So these are serine proteases, and they are going to induce the apoptosis. So these are serine proteases which will induce apoptosis, and this is how the cell will be killed. Okay, so if not direct cytotoxicity, natural killer cell, they can activate the death receptor pathway. If you don't know about it, I have discussed about it in detail in the apoptosis lecture in cancer biology. So the death receptor pathway is the extrinsic pathway for apoptosis, for the programmed cell death. So this is also mediated by natural killer cells. So the natural killer cells, they activate the death receptors present over the target cells, and that is how they are going to mediate the cytotoxicity. This is the natural killer cells. It has a few receptors over it, such as the FasL ligand, or which is also referred as CD95L. Okay, so this is actually a receptor for the, this is going to go and bind to the death receptors. Okay, so this is one of them. Then there are the TREL receptor, the TRELs molecules which are which are over there on the natural killer cells. Okay, so those particular proteins, they will be those particular proteins, they will bind to the death receptor, sorry, death receptors present over the target cells. So the target cell has the death receptor called FAS. It has TREL receptors. Okay. Okay, so these are particularly present over the target cells, the cells of our body, and once they get activated, once this interaction happens, then they will start the intracellular signaling cascade, which will lead to the extrinsic pathway of the apoptosis. Okay, if you don't know about that, I have discussed that in great details in that particular lecture about apoptosis, as well as the how tumor cells they are going to evade apoptosis in the cancer biology. So please refer to that lecture. So these are the two ways by which natural killer cells can mediate the killing. And there's a third way, which is really important and which is really specific to natural killer cells, and that is referred as antibody dependent cell mediated cytotoxicity, ADCC. Antibody mediated or antibody dependent cell mediated cytotoxicity. So in this particular phenomena, this way is very particular to natural killer cells. So in this way of killing the target cell, natural killer cells actually has the receptors, the CD16s, which are going to bind to the antibodies, and those antibody they are bound to already the target cell. So what happens is that antibody can go and bind to certain cells. So those antibody labeled or antibody coated target cells, they will be recognized by the natural killer cells. So natural killer cells has certain receptors such as CD16, okay, CD16, which are going to bind to the FC region of the antibodies. And let's say that there is this particular target cell which is being labeled with the antibodies. So I am referring it as antibody coated target cell. Okay, so this antibody coated target cell has antibodies which are present over it, which are bound to the antigens to or the proteins or the cell surface markers of this particular cell. So this will be received by those receptors present over the natural killer cell. So they will interact with those receptors, and this interaction, the antibody and the CD16 interaction. So let's assume that this is IgG antibody. So the interaction between those two will result into activation of natural killer cell, which will trigger the degranulation of the natural killer cell, releasing its perforin and granzyme, and that will result into killing the antibody tagged cells. This will result into killing this particular cell. So this is also a really really important way of killing the cells which are being infected. And the factors that can affect this antibody dependent cellular cytotoxicity, they are the factors such as density and the stability of this particular antigen over the target cell, that will affect this ADCC efficiency. Then the antibody affinity to the receptors present over natural killer cells, means the interaction efficiency of those IgG antibody and CD16, that will also affect this particular phenomena, the cytotoxicity. So this depends on multiple factors such as those that I discussed right now. So depending upon that, the natural killer cell activity will be variable, right? Whether it is functioning properly or not, and we can assess them. So there are particular assays for measuring the natural killer cell cytotoxic abilities, whether they are killing the target cells or not, how efficient they are. So for measuring such measures, so for measuring such things, there exists some assays, and these are referred as the NK cell activity measuring assays. So in previous days, we used to use chromium, the radioactive chromium, for measuring the activity of natural killer cells. That used to be referred as chromium release assay. Okay, so this chromium release assay, it used to give us the index of efficiency of the natural killer cell mediated cytotoxicity. So how this assay works? So we actually provide the target cells with the chromium salts. So chromium, which is radioactive, so the chromium 51 is a radioactive chromium. So we used to provide the chromium salts such as Na2CrO, CrO4. Okay, so in this, the medium having those radioactive chromium, in those medium, the cells were grown, and those cells, they are now going to serve as the target for the natural killer cells. So those target cells has this, radioactive chromium, which is inside the cell. Means this chromium, this radioactive chromium, is intracellular. So once the natural killer cell is going to, bind to this target cell, and it is going to kill this target cell. So the natural killer cell, when it will kill the target cell, this radioactive chromium, which was previously inside the cell, it will be released into the extracellular environment, right? So this will result into chromium release, and this radioactive chromium release is going to give us an idea about the efficiency of killing of the target cells by the natural killer cells. So the percentage of cytotoxicity will be measured, and that is going to give us an idea about how much efficient that natural killer cells are, right? So besides so this is, due to the radioactivity, this procedure is very tough, and it is not that flexible, and it is not efficient in and it is time consuming. So nowadays we use the lactate dehydrogenase release assay. So there is an enzyme which is present in this inside the cells, that is referred as lactate dehydrogenase, that is going to convert the lactate into the lactic acid into the pyruvic acid. So this lactate dehydrogenase release assay also does the same job for us. Okay, so in this particular assay, when the lactate dehydrogenase works and it forms its products, that products will be linked with certain other reaction that is going to result into the that is going to result into breaking down the chromogenic product. So what happens is that lactate, okay, the lactate will be converted into the pyruvate with the help of this lactic acid dehydrogenase, right? So this particular reaction is being catalyzed by this particular enzyme. So this, when this particular enzyme will work on this lactic acid, it is going to release the pyruvate. So what happens is that when natural killer cells kill the target cell, they are going to release this particular enzyme in the supernatant. And when this supernatant is going to have this particular enzyme, we will provide it the substrate, and this substrate will be this NADH, which is being produced. It will be coupled with the chromogenic substances, such as the tetrazolium salts. So it will be coupled with the reactions involving the tetrazolium salts, and that is going to give us the color. So if to state simply, the more is the LDH in the particular supernatant, more will be the intensity of the color. Means, and when will the LDH concentration will be more in the supernatant? When the cells are being killed and they are releasing the LDH, because this particular enzyme is not present in that particular, in that particular sample. This is actually inside the cell. So if this is the tray and these are the cells, and here particularly we have the substrate, the lactate, on which the lactate dehydrogenase is going to work. So that particular enzyme is inside the cells, so it cannot work on the substrate which is present outside the cells, right? So for that we need the release of this particular enzyme in the extracellular environment, and that is only done when those cells will be killed. And that is why this will tell us about the efficiency of the natural killer cells. So the more is the release of this dehydrogenase, more will be the intensity of the color, and this will refer that the natural killer cells are very effective and their cytotoxic activity is quite high. Okay, so I hope you have got an idea about how the natural killer cells, they are going to mediate the killing of the target cells. Okay, so they use those different methods for killing, and we also discussed about different types of the natural killer cells and how to recognize them using the surface markers. So with this, I'm done with this lecture. There is also one more type of the natural killer cells which actually has functions of both T lymphocytes as well as the natural killer cells, which is referred as natural killer T cells. So we will discuss about this natural killer T cells in next lecture. Till then, see you. Bye bye.